Good morning, everyone. Today we are going to study about the structure of the salivary gland. This is the general structure of the salivary gland, which includes the parenchyma and the connective tissue capsule. The parenchyma consists of mainly the terminal secretory unit, which is called the acini, and the ductal system. Then the connective tissue capsule, which comprises of the blood, the lymph vessels, and the nerves. So first we will have a glance on the parenchyma, which consists of the acini and the ductal system. When we take the parotid gland as an example, we can see that if it is cut into sections, there's an arrow mark which shows the cut section of the parotid gland. So we can see the ducts and the acini patterns can be seen. Then the second one is the connective tissue capsule, which comprises of the blood, lymph vessels, and the nerves. So this is the connective tissue capsule, which can be seen in the outermost layer. That is, in this diagram, you can see the capsule. This is the outermost capsule you can see. And we can see that the parotid gland, it is divided into lobes. And within that, we can be divided into lobules also. So via these connective tissue capsules, the blood vessels, the lymph vessels, and the nerves provide nutrition to the parotid gland. This is the light microscope structure of the salivary gland. So the salivary gland consists of mainly a series of branch ducts you can see. So which terminates in spherical acini. That is, these are the end pieces, which has a similar to that of grapes you can see. The grape stem, which has the similar to that of a ductal system, and this is the terminal end pieces. That is the grapes. Each grape pieces can be similar to that of an acini you can see. So this acini can be of two types. That is, it can be a serous type and a mucus type. So next, a basement membrane. So as we all know, the basement membrane is that the cells in which the acini rest, or whatever the cells rest on a membrane, it's called as the basement membrane. So here the cells in the acini also rest on a membrane which is called the basement membrane. As you can see, these are the acini cells which rest on a basement membrane. And this basement membrane is also arranged in a single layer. And we can see the intercellular spaces of the apical end of the cells are separated from the lumen by certain types of junctional complexes. So these complexes can be either it can be tight, so which is called the zona occludens, and it can be of an intermediate junction, which is called the zonal adherens. And the last type, it is the desmosomes, which is called as the macular adherens. So the function of this junctional complex, it is mainly it holds the cell together, and they also regulate the permeability. The tight junctions, they helps in sealing and they control the influx of the ions, and and it also maintains the cell polarity and helps in the tissue homeostasis. So the main tight junctional proteins are the claudins, occludins, and the junctional adhesion molecules. So next we will see about the ductal system. So we have the ductal system, such as first one is the main excretory duct. You have the main excretory duct. This is the main excretory ducts, which divides into smaller interlobular ducts, which divides lobes. As we have already seen in the previous slides, what is the difference between lobes and lobules we have seen. So first it will be lobes. Then comes the lobules. So this main secretory ducts, it divides this into interlobar, and then comes the interlobular duct. So the predominant intralobular duct component is mainly the striated ducts. First, this is the striated ducts. Then this is your intercalated duct. This intercalated ducts, which connects the striated duct to the acini. Then this is the end pieces that we see in that grape. This is the end pieces, or it's called as acini. So this lumen of this end pieces, it is continuous with that of the intercalated duct lumen. So sometimes in some glands, there will be small extensions of these lumens between the acini. So this is called as intercellular canaliculi. They are found between the adjacent secretory cells. So these intercellular canaliculi can also extend to the base of the cells also. It can extend up to the base of the cells also. So we'll see the acini. We have already discussed that the acini are of mainly two types. That is your serous acini and the mucous acini. So the acini in a Latin word, it is means a berry or it is a grape. So it is the main functional unit of a salivary gland. It mainly comprises of mainly round or tubular collections of secretory cells. So which forms the secretory unit of the salivary gland. So the serous salivary glands may composed mainly of the serous acini, and the mucous glands are mostly mucus. And another type you have a mixed salivary gland. So there you can see both your mucous acini and the serous acini, and they are arranged in a form called as demulunes, you can see. So sometimes there's a mucous acini that we already seen, that is a demulunes. We have seen the demulunes. So demulunes, that is mucous acini, will be bonnet shaped or it will be crescent shaped, and the covering which is made of a serous cells. That is in this picture you can see that this is a mucous cells which is covered by a crescent shape, that is a serous cell. So this is also called as a demulule. So the presence of the demulules, it is mainly a questionable thing. Some says that it is seen in the mixed salivary gland, and some they are saying that it is mainly due to the artifact, that means a fault during the tissue preparations. So first, this is the serous cells we can see. So as we can see in the picture, the serous cells is mainly pyramidal cells. It has a broad base on the basement membrane, and you have an apex which faces the lumen. So we have seen this is the lumen. So this is the apex and this is the base of the cell. So it is the serous, this is the apex, and they have a broad basal cells. And here itself we have seen the basement membrane. So the nucleus, it is almost spherical in shape and it is placed at the basal region. Then this is the apical cytoplasm. They consist of so many secretory granules can be seen. These granules are approximately 1 millimeter in diameter and they're closely opposed to each other. And these granules are mainly zymogen granules and they are formed by glycosylated proteins which are released into the vacuoles. So again, this is the acini. This is one serous acini. We can see these are the zymogen granules and this is the basal portion, that this is the apical portion, and you can see the spherical size of the nucleus, and this is your basement membrane. So in an acini, you can see 8 to 12 serous cells can be seen. They are arranged around a small lumen. So in the right pictures, you can see a mucous acini. Mucous acini, you have a large lumen, you can see. See here it is roughly round in shape, while mucus, it is somewhat, it is oval in shape, you can see. Then this is the central lumen, which is extends between the secretory cells. It can extend between the secretory cells also, that we will see in the next slide. And that is known as the intercellular canaliculi. So this is the lumen which extends here. This is called as the intercellular canaliculi. We can see this. And next, the serous acini, which is comparatively smaller in size when compared to the mucous acini, and it has less number of cells and have a relatively smaller lumens, you can see. Then the electron microscopy pictures of the serous cells, they also show so many organelles which are mainly actively take part in protein synthesis. So the plasma membrane, they have specializations. Luminal cells, we have seen intercellular canaliculi. Then lateral surface have occasional folds can be seen. Basal surface, regular folds. In the basal surface, they have regular folds. Mainly the foldings are there due to increase the surface area. Serous cells as well as the mucous cells are joined together by the intercellular junction. So in these pictures, we can see. So this is a pyramidal shaped cells. You can see this is the basal side and this is the apical side. Basal cells, you can see so many infoldings you can see in the basal side, and also on the few on the lateral sides also you can see. So these infoldings are mainly due to increase the areas. And next, this is the lumen. You have see smaller lumen, we have already said. The lumen which extends between the cells also. So this is called as the intercellular canaliculi. Then somewhat oval shaped or round shaped nucleus, and it is seen at the basal side of the cell. Then these are the zymogen granules we can see in the apical cytoplasm. And between the cells, you can see the junctions. That is the cell junctions you can see. And the numerous organelles you can see this is a mitochondria, this is a Golgi apparatus, this is endoplasmic reticulum, and this is a Golgi apparatus you can see. So the basal cytoplasm is with a rough endoplasmic reticulum you can see, large Golgi complex also you can see, just apical or lateral to the nucleus. Granules you have seen on the apical cytoplasm, mitochondria and lysosomes can also be seen, which helps to destroy the foreign materials and worn out cell organelles you can see. So this is a H&T picture of Serous acini. You can see it is mostly eosinophilic stains you can see. So the most eosinophilic it is mainly due to the presence of the granules. This granules at the apical cytoplasm which makes it more eosinophilic. And this is the basic protein synthesis we have already learned in the basic science part, that you have already learned how a protein synthesis occurs. In the nucleus, you have your DNA, and the protein synthesis will be taking place in the nuclei. The messenger RNA, the ribosomes, they carries the messages from there, and it translates into the rough endoplasmic reticulum. It has cis part and trans part, which gets modified and transferred in the Golgi complex, then from there to the vacuoles, and they secrete to the process called as exocytosis. And this is the mucus cells. So mucus cells, it is mainly for the secretions of the secretory products. You can see so many mucus droplets you can see. So it consists of larger amounts of secretory products. These secretory products, they are rich in carbohydrates at the apical cytoplasm. Then they are a relatively larger lumen when compared to that of your serous. Then the mucus, it is also larger in size when compared to that of the serous also. Then here the nucleus is overall flattened shaped. It is also just located above the basal membrane. Then rough endoplasmic reticulum, a narrow band of cytoplasm can be seen limited along the base and the lateral borders of the cell. Mitochondria and organelles also seen. Then Golgi apparatus can be seen. So this is again, it is a pyramidal shape. This is the apex, this is the apical part, and this is the basal part, and you can see this is the lumen part. So you can see the nucleus, it is somewhat an oval, or it is flattened in shape, and it is sandwiched between your rough endoplasmic reticulum and the Golgi apparatus, and you can see so many mucus droplets can be seen. Then the secretions of mucus droplets, that is when a single droplet is discharged, its limiting membrane, it gets fuses with the apical plasma membrane and result in a single droplet, and then it gets discharged with the membrane intact surrounding it. So during this discharge, sometimes the apical cytoplasm may not be sealing itself off. So it seems to be that entire mass of the mucus may be spilled into the lumen. And this is the H&T sections. And in the H&T sections, you can see some clear cells you can see. In the serous, you have seen it is somewhat eosinophilic, but here it is somewhat clear. That is, the space seems to be empty. And these are mainly the mucins, which does not take the states, and it consists of heavily glycosylated proteins. So for viewing these mucin types, you have to take special stains such as music-carmine. This is the music-carmine stains, or PAS stain, that is the periodic acid chip stain, or Alcian blue stains. This will be specific colors to this mucin. So for an example, Alcian blue, it is blue in color, you can see here. So last one is the mixed acini, that we have already said that it is a mixed, means it consists of both your serous and the mucus, which can be arranged in a crescent-shaped structure. The serous cells will be arranged in a crescent shape to that of the mucus acini, and it's called as a demilunes. Demilunes means it is half moon. So the demilunes, it is called as the demilune of Chianesi. And this is the mixed acini. You can see this is a mucus acini in which your serous, which is seen as a serous demilunes, can be seen as a crescent shape, you can see. So this is the basic differences between the serous and the mucus acini in each view, you can see. The light microscope, the general differences between the mucus and the serous, you can see. And the mucus acini and the serous acini, the electron microscopic view also you can see. So that we have already that pyramidal shape, the nucleus differences. Then the apical portion, zymogen granules can be seen in serous, here empty. Then H&T stain differences can be seen, and what all the content of the granules can be seen. The staining intake you can see for a mucus individual cells difference you can see. And when a group of cells, it is called as the acini. So the general shape, it is elliptical and spherical, and the lumen shapes you can see, and the intercellular canaliculi, and the secretion types and the product types can be the major differences between the acini. And the electron microscope also, the granule differences can be seen and the organelle differences can be seen. So the another type of cells, it is the myoepithelial cells. So you can see the myoepithelial cells, it is a specific type of cells. So spider-like cells or stellate shaped cells you can see around the acini. These are called as the myoepithelial cells.